Okay, guys, let's do something. So now I feel like the class is even smaller. You guys all know your grades, and most of you did really well. So either you're not coming to class, those are the people who are listening. Either you're not coming to class because you did so well you don't need to come to class, or you've given up. I'm not giving back the exams in class because there's so many of you, so just grab them at recitation, and that way you can ask whatever questions you need to. But people did really well overall. I forgot to calculate the average. Sorry. I'm guessing, I checked the averages among the 3 different sections just to make sure that they were almost equal, and they ranged between 77 and 85, which is pretty amazing. So clearly the 77 got pushed up a little bit. But yeah, so good job everyone. Okay, cell cycle. Right, so exam 1 grades are posted. Hopefully you saw that. Wednesday recitation, you're going to make up the snow day material this week, so be ready. Exam 2 is the week of 4-1. There's been a lot of confusion about that because those of you who printed out the syllabus and never looked again have an old syllabus. So make sure you look at the revised syllabus on Blackboard. Okay, so we're going to start. Does anyone have questions or comments or concerns? Because I just heard a lot of homework. Nope. Is everyone happy? Okay. Okay, so there was a question about this weekend's quiz. There's always something, isn't there? So there was this question, how many DNA polymerases are present on a bacterial chromosome during replication? And I got a handful. And by handful, I mean only like 3 or 4, which isn't so bad. But I still feel like if I had that question multiple times, then I should address it in class. People were very confused about this question. And I think for whatever reason, people read it as you wanted to know how many types of DNA polymerase were in bacteria. I'm guessing. And that's not what I'm asking, right? How many DNA polymerases are present on a bacterial chromosome during DNA replication? Okay, that is the question. So remember, bacteria have one origin, right? One origin, which means that they're going to have 2 replication forks, right? Are we in agreement? And that means each replication fork is going to look like this. And so here's 1 and 2 times 2 is 4. And so that's how I got to 4. I'm trying to get you guys to think about what it looks like at the replication fork. Okay? I don't know if the wording just threw people off, but it looks very straightforward. Present on, because they actually are binding to the chromosome. Yes? Can you explain that? Sure. Can I explain that again? I will, because probably more people than just one want that. Right? So you've got one origin in bacteria, right? Everyone follows that. And so that's what you see here. And so as it opens, you're going to have one replication bubble, which contains 2 replication forks, one moving in either direction, right? And at each replication fork, you have your leading and lagging strand being replicated, right? So this machine here, sometimes talked about, referred to as the DNA polymerase holoenzyme, contains 2 DNA polymerase enzymes. So each replication fork contains 2 DNA polymerases, and there are 2 replication forks, so it's 4. So could I have asked this question about eukaryotic chromosomes and been fair in any way? No, that would have been mean, right? Bacteria is easy, 1, so it's simple math. I'm sure there are people who have calculated the number of origins in eukaryotic cells, but it's a huge number and doesn't really illustrate much of anything except for my desire to make you do lots of math. Yes?
So if I was to 4, I understand that there's one going in the leading strand and one going in the lagging strand.
And that's 1 replication fork, but you have 2 replication forks. We've got 1 going to the left, 1 going to the right. One's going east, one's going west. And so people always forget that because in books, they tend to just show one. Of course, because for simplicity, they're showing one a lot of the time because they're showing that detailed picture with all the different proteins in there. But I want you to really understand it's a replication bubble with 2 replication forks, one going each way. And each strand needs a polymerase to copy it, right? Yes?
So I guess why I'm confused is it like two double-stranded DNA like together makes one chromosome? Because when the way I look at it, it looks like one double-stranded DNA is opening, and that would need a lagging strand. You know, because you have 2 separate DNA strands, and then those
Right.
are being
Right, so you've got one double helix. Here's your parental DNA, right here, the yellow. And it opens up and becomes 2, right? You have one double helix, and each strand is copied.
Yeah.
Each strand is copied. Remember, you've got double-stranded DNA. Each strand is copied. You need a leading strand and a lagging strand, right? But there's 2 replication forks, one on each side. So one goes to the east, the other goes to the west, and they meet at the bottom. Yes?
How do they make sure that if they meet at the bottom, isn't there any DNA that's still needs to be?
They meet, they replicate until they collide. Yep.
And they just kind of merge together.
They just merge together. And so the question was, how do they meet? They just kind of merge together, and that's also what happens in eukaryotic cells, where you have all of these origins all replicating at the same time until they merge. And we're going to learn about that. Any other questions about this? It seems to throw people off, and all I could think was people were reading it wrong. Hopefully. Yes?
So what fixes the errors in bacteria?
What fixes the errors? We will get to those. Yeah, we're not going to worry about that just now. I just want to explain this quiz question. We're going to do a whole bunch of DNA repair. Yeah. Okay, any other questions? I want this to be clear because if you don't understand this concept, then I'm failing you in actually the whole biology department. Yes. 4 polymerases, right? You've got 1, see the green olive? Who likes martinis? Who likes them dirty? I'm not trying to be gross. Green olives, martinis, they're dirty when you pour the olive juice in. Anyways, green olive, each green olive is a DNA polymerase. So how many do you see there? 2, right? 2 green olives. That's 1 replication fork. But there's 2 because, as you can see here, it goes each way. You're missing it. Okay, come see me. Come talk to me. Okay, questions? Okay, this is just an important one to make sure everyone gets. Okay, so the answer was D. Well, the answer was 4. I have it so it randomly jumbles, so it could have been anything for you. Okay, let's move on. So far we've been studying cell cycle. We study cell cycle regulation, the cycles, the CDKs, the CKIs, the kinases, the phosphatases, all that stuff that regulate whether or not you're going to go forward into S phase or M phase or whatever. We talked about checkpoints briefly, right? Because you don't want to move into S phase if everything's all messed up. DNA polymerase, we introduced that, just the enzyme itself, the fact, we looked at its structure, we talked about what it does, we talked about how it has proofreading capabilities, and then we talked about enzymes that are involved in DNA replication. We spoke about polymerase, helicase, primase, ligase, and topoisomerase. So that brings us to the mechanism of DNA replication, right? And we started that on Friday. Initiation, priming, leading lagging strand synthesis, right? This is the replication fork formation, and it goes until it terminates. Okay, so let's talk about initiation. It starts with the binding of initiator proteins to this origin, and origins are made up of 2 main elements, right? An AT-rich DNA element and an initiator binding site. And they're loaded and they just kind of sit there until they're activated in some way. And those things are going to differ whether or not you're a bacteria or a eukaryotic cell. In eukaryotic cells, they're activated through cyclins, and ORC, the origin recognition complex, is recruited to origins with hyperacetylated tails, so you're going to have a more open DNA structure. So let's get a little bit more detailed and talk about bacteria. That's the simplest model, and where most of the research was done initially. And it's fairly simple. It looks complicated, but they're just basically 1, 2, 3 main parts, I guess. So DNA A, which is the origin. Well, actually, let me see. I think I might have it in a better. Okay, well, we'll do it a few times to make sure everyone gets it. DNA A binds to this DNA binding region. So this is DNA A binding region upstream or downstream from the AT-rich region, and it actually causes the DNA to change its shape, and that helps with it opening up, and so it opens up, and then DNA C comes along with DNA B, and this loads the helicase onto the DNA, and then disassembles, and then you get other things. You get recruited, such as primase, and then you have establishment of your replication forks. So this origin is really simple. It has 2 major regions, right? This initiator binding site that attracts the initiator proteins, and this AT-rich region, which is easy to open. Why do I say it's easy to open? Does anyone remember why AT-rich is easier to open? Lots of people. Who do I never hear from? Yeah.
Less hydrogen bonds.
Less hydrogen bonds, absolutely, right. So, and this is something that you should just always know, because let's say you have to design PCR primers. It's an important thing to understand with that, because it has to do with how well your PCR primer is going to bind to your template. So right, so your AT has 2 hydrogen bonds and your GC has 3, so it's stronger interaction, right? So why is it helpful for your origin to have an AT-rich region? What if it was GC-rich? What would happen? It would take, I heard it would take more effort. So it would be tighter bonding. It would be harder to open, right? You would want your origin to open easily, right? Makes sense. Okay, so the basic process of replication fork initiation, it's fundamentally the same in bacteria and eukaryotes. Slightly more complicated in eukaryotes. But the basics are, you have your replication origin, then you get the local opening of your DNA helix, primer synthesis, leading strand synthesis, and then lagging strand synthesis, and then you have your replication forks going in both different directions. See your 2 replication forks? Yes?
If bacteria chromosomes are circular, why does it need to be lagging strand?
Because you have to copy both strands of DNA. So the question was, why does there need to be a lagging strand? And we're going to go into that with more detail. Thank you for that question, because it's an important one. Each replication fork has a leading strand where you've got your primer that synthesizes and you get direct replication. And then you're going to have your lagging strand because you need to go in both ways. Okay? I guess you could just start in one way and go all the way around, and the other way and go all the way around. Seems more time consuming. It would double the time that it would take, right? If you've got 2 that go this way, or you have 1 that goes all the way around, and 1 that goes all the way around.
Going like ratcheting, it's lagging strand. So it is still taking time.
Okay, so for whatever reason, bacteria have decided to evolve this mechanism and keep it. So I'll say that that's efficient in some way. If it was very inefficient, I'm guessing it wouldn't have survived. Right, so this replication fork initiation is what causes the replication bubble. So those are two terms that you need to understand. Replication fork, which is the thing that kind of looks like a fork, and the bubble, which kind of looks like the bubble. Okay? Yes?
So bubble is the whole thing?
Yep, so in bacteria you'll have one replication bubble with two replication forks, and in eukaryotic cells you'll have lots of replication bubbles. And it depends on how many origins you have. Yes?
How did the RNA primers get moved?
How do the primers get removed? We will get there. Oh, we will get there. Yep. How do the primers get removed? Okay. So, DNA replication in bacteria. One origin, two replication forks, one replication bubble, two DNA polymerase enzymes at each replication fork, right? One leading, one lagging, one leading, one lagging. 4 DNA polymerase enzymes per genome per replication cycle. That's how it works out. I know I'm nailing this one in, but I'm guessing people have confusion. Okay, so, and you end up with 2 circular DNA molecules. Okay, so let's get a little bit more detailed, right? So the major players in bacteria are DNA A, which is the initiator, DNA B, which is a helicase, and DNA G, which is a primase. And they occur in the order of initiator, helicase, primase, and then DNA polymerase combined and replicate the DNA. And if you think you don't need to memorize this order, because if you know what each enzyme does, it's pretty logical about what comes when. If you just try to blindly memorize it, then you'll make dumb mistakes. Right, so you've got your origin, which is this AT-rich sequence, and then also this initiator binding region. And here it's showing it a little bit differently. The initiator proteins, also known as DNA A, will bind and distort the helix. And that helps with loading of helicase and opening up the DNA. So there's a helicase loading protein that brings the helicase on and helps to load it onto the open DNA. So there you see your helicase on your DNA. And then it opens the DNA, and this allows the entry of the primase, which is DNA G. And primase is going to make your primer, and then that allows polymerase to come and add on to that primer. And that's how it begins. And so you'll get leading strand first, and then you'll do that for each of the 4 strands that need to be copied. 4 strands, well, think about it as 2 replication forks, each has 2. Okay? The helicase loading protein actually inhibits the helicase activity until it's loaded onto DNA, so that prevents the helicase from going to the wrong place and binding and doing the wrong thing. So it's a level of regulation. Yeah?
So does DNA primase find this specific DNA strands?
Does DNA primase bind to specific DNA sequences? I believe it more has to do with interaction with helicase. And you'll see as we start to learn about DNA replication in gory detail and recitation this week, that this whole complex interacts. It's a bunch of proteins that are interacting together. Yeah, and primase is one of them. Yes?
In some ways, telomerase is the only reverse transcribing protein in humans. What does primase do?
Primase makes an RNA primer.
So it's just like a short.
Makes a little short primase. So the question is, what is primase doing? And it makes a little short bit of RNA that binds to the DNA template, and that's the OH group that the new nucleotides are added onto. and then it's removed later. Telomerase has its own RNA primer in it. It's part of the enzyme. So it's reverse transcribing. Okay, think about it a little and then we'll reconvene. Okay, any questions? Who loves DNA? Okay, that's good. Okay, let's move on. So in bacteria, initiation is regulated by availability of the origin for interaction with initiator proteins. It's probably also regulated by abundance of initiator proteins, but I don't have that data, but I'm sure it's there. But the thing that they talked about in the book is how the methylation status of the origin determines whether or not you will get initiation. The bacterial origin needs to be fully methylated in order to bind to these origin recognition, these initiator proteins. And so if you think about it, when you replicate your DNA, you're going to have heavy methylated DNA because your parental strand will be methylated and your new one won't. And then it'll take a little time, and then slowly the enzymes will come and methylate the DNA, the new DNA, and then you have fully methylated chromosomes. And so that's a way that the bacteria regulates initiation. So in essence, there's this refractory period after replication where you cannot initiate replication again. Okay? And so this is what this figure is showing. Here's your fully methylated origin. It looks like a cone, kind of. And then you get replication. So you've got your replication bubble here. Notice your new DNA is not methylated. And so it actually cannot be bound again by the initiator proteins, and DNA replication cannot be initiated again. And then after a while you'll get complete methylation, and that will allow replication to occur again if needed. So regulation is based on environmental conditions, nutrient availability, but also based on the ability of the origin to be fully methylated so that the initiator proteins combine. Yes? We're talking about bacteria. So the question was, why methylated? Doesn't methylation inhibit things? So all the stuff that we talked about, and thank you so much for bringing this up, because it's a really good way, a good time to kind of think about what is methylation doing in bacteria versus eukaryotic cells. And don't forget, methylation in eukaryotic cells is involved in regulation of gene expression. So whether or not transcription is going to happen. And yes, methylation of cytosines will inhibit RNA polymerase from binding and inhibit recruitment of all the enzymes required for gene expression. Yes. In bacteria, though, a methylated origin is required for interaction with initiator proteins. And if you think about it, think about what methylation does. What's it doing? It's changing the shape of something. That's all it's doing. So that's why I caution you to always think of methylation as inhibitory, because sometimes it's not. And in bacteria, it does a completely different thing anyways. But you just need to think of these modifications as changing the shape of something, and it's going to affect interaction based on whatever needs to bind, right? Just like phosphorylation, sometimes will encourage something to happen, sometimes will inhibit something. Same thing with methylation. Sometimes it will encourage gene expression, sometimes it will inhibit. And in this case, methylation is required for binding of initiator proteins. So that's the shape that those initiator proteins need. Make sense? Yes?
So, just want to clarify. So, in bacteria, is methylation also always inherited to the next generation?
In bacteria, is methylation inherited to next generation? Yes, because in bacteria, the genomes are always methylated.
Okay, so
It's a whole different story. It's like a completely different thing. Don't think of, I don't know that much about gene expression in bacteria, but I think in terms of regulation of gene expression, think of that in terms of eukaryotic cells and methylation and epigenetics and all that, right? And then in bacteria, think of it as just there are different mechanisms. Yeah. More questions? Though sometimes it's really easy to get confused when you're thinking about replication and transcription and translation and all these things, and you need to start to get that big picture of what's the goal here. The goal here is to replicate the genome. Okay, questions? Okay. So I forgot to ask a very important question back here. So what would happen if these proteins were recruited in the wrong order? Or could that even happen? Why is the order so logical? I said it was really logical. Here, I'm gonna. Kristen Sessa.
They build off of each other. They obviously not work.
Right, so they build off each other. Absolutely. One is always going to be required for the next step. So if the wrong thing binds at the wrong time, it's not going to work. And so I want you guys to really think about that when you're studying. Is if there's no initiator protein, you're not going to get the right conformation of the DNA for it to open up and for the helicase to be loaded. If there's no helicase loaded, you're not going to open your DNA. If there's no open DNA, then you can't get your primase to come in and make your primer. If there's no primase and there's no primer, then DNA polymerase can't polymerize DNA off of that. Yes?
Where does topoisomerase fall into this?
Topoisomerase is involved in reducing the torsional stress. So as your replication fork is opening, all the DNA that hasn't been opened yet starts to become stressed because you could think of it as winding tighter and tighter. That's where it comes in to relieve that stress. So it's way ahead.
So it comes before all of these, or?
No. Well, no. So you have to think to yourself, what does topoisomerase do? Right? So it relieves torsional stress that comes from opening the double helix. Right? So the double helix needs to be open first in order to need to relieve stress. Otherwise, there's no reason to need it. So what's happening is it's probably many bases ahead of the replication fork and kind of stays ahead and senses the torsional stress in some ways and knows how to. I don't know the exact regulation of the mechanism of when it chooses to work, but it's ahead of the replication fork. Yes?
Do bacteria utilize exonuclease?
Huh?
Do bacteria utilize exonuclease?
Do bacteria utilize exonuclease? I'm sure they must. You mean, does the DNA polymerase in bacteria have exonuclease activity?
Sure.
Yeah, I believe so. Yeah. Yep.
So what is this binding protein? Is it different from
Okay, so the question is the difference between single-stranded binding proteins and topoisomerase, right? So single-stranded binding proteins do exactly what they say they do. They bind single-stranded DNA. So once the helicase opens up the DNA, those single-stranded binding proteins come in and kind of cover the single-strand DNA and prevent the DNA from closing up again because that double helix is such a strong, stable structure. It wants to come back together. Topoisomerase is going to deal with taking the tension out of the double helix that results from helicase activity opening up the double strand of DNA. Make sense? Okay. Yes.
So bacteria would utilize topoisomerase as well.
I don't see why not. Bacteria should need topoisomerase, right? Because they've got a double helix that needs to be opened, and you've got two replication forks, so there's bound to be a lot of torsional stress in that in between point. Yeah. Questions? I'm getting great questions. Yes.
On the next slide, the refractory period is just to allow time for the fully methylated origins to develop, right?
The refractory period is the period of time before the methylated origins are present. Yeah. So it's not about allowing for it, it's actually a way to slow down initiation. So you don't have this continuous initiation, right? Because otherwise, what's to stop initiator proteins from constantly binding to origins and replicating? But what if you're still replicating? I mean, you know what I mean?
Yeah.
Yeah. Yes.
Doesn't the hemimethylation have something to do with proofreading ability?
Right, so we talked about proofreading too before, right? So hemimethylation is also involved in proofreading. Right, there are many functions for a lot of these things. So we learned a few weeks ago about this, right? How in bacteria, your old DNA is methylated and your new DNA is not. And so that's how the bacteria knows what the correct sequence is and knows which one to base the correction on when you're proofreading and correcting incorrect, adding correct bases. Yeah. How are we doing? Yes.
So, as the DNA is being replicated, does the initiator come up or does it stay on it?
So as DNA is being replicated, does the initiator protein stay bound to the origin? I would say it must. So this is your initiator proteins here. I'm thinking they must come off because they'd be inhibitory to replication. They're bound. They bind the DNA, and it needs to be accessible to whole grains. How are we doing? Is everyone having fun? It's Monday morning after the time change. Yes.
So is the new strand methylated after it's been proofreaded?
Is the new strand methylated after it's been proofread?
Sorry.
It's okay. I have young children, so my English is. I used to work in a completely non-English speaking lab, so my English started to really deteriorate. It was like 10 years of non-native English speakers. And I don't know what happened. My English just deteriorated, and then now I have like toddlers, and so I almost didn't catch that. Okay, your question is methylation. Does that occur after proofreading? That would be logical. That's a logical assumption. More questions? Okay, let's go. Okay, so let's go. So that was all bacteria. Simple, simple, simple. In eukaryotic chromosomes, there are multiple origins, and we did a paper on that last week, so it's all pretty clear to you guys. But let's talk a little bit more about it. So the initial experiment where they discovered that there were multiple origins, they used radioactive thymidine, so that's the T, that's put in your DNA during replication. They used that to label cells in culture. And then they lysed the cells, and they would stretch the DNA out on the slide, and then they would detect origins by autoradiography. And so what would end up happening in the autoradiography, that had something to do with some photography emulsion, I don't know. But basically what would happen is you would get these silver grains that would show up where the radioactivity was. And because remember, you're looking at replication bubbles. Okay? So you would label for a short period of time, and then you would expose it to film, and you would only see the newly replicated DNA, because that's the only place where the radioactive thymidine is being incorporated. And if you do it over a different time period, you would see expansion of these replication bubbles. So you would start just seeing this, and then next time it would be longer, because more DNA would have replicated. And it was reproducible because the origins are not random. They're located in certain places and in certain clusters. And these are the first experiments that were done to study this. What blew my mind when I was reading about this was that to get this data, they had to expose their slides, so they stretched their DNA out on a slide and then treat it with this photographic emulsion and then expose it to X-ray film for months. Months. Can you imagine doing an experiment and having to wait for months to find out if it works? Who's worked in a lab? What's your success rate of experiments? How much time is the experiment working and how much of the time is it not working?
It's not working.
Right, so there's about a 1 in 10. Like of every 10 experiments, I think from undergrad all the way through postdoc, you're going to find about a 1 in 10 success rate. Well, you know, science isn't easy, is it? Can you imagine you wait a month and then it doesn't work? Sorry, I saw that and I was like, no way, man. Thank God we're in modern day, because this was a long time ago. Now we have much cooler, faster ways to do things. But still, what did I do? I used to do pulse chase. That's where you label protein and you can analyze the half-life of a protein, so how quickly it gets degraded. And I would have to expose for like a weekend, and that was torturous. Like a weekend. Couldn't handle that. Anyways, enough about me and my occupations. Okay, so this was what we did last week in recitation, right? So this is another way that people did, that people studied origins. And this is way more, so there they were just looking at, oh look, there's a small replication bubble, now it's larger, and there's many of them. Now what they do is they're actually looking at locations of replication origins by hybridizing the DNA to the newly replicated DNA to microarrays. And you can start to study it that way. And I'm not going to go into it because you guys studied it. We already talked about it last week, but I just wanted to make you realize that something you read does connect to lecture. Do you guys ever wonder why we're reading what we're reading in recitation? I hope not. No, it means I'm doing a bad job. Okay, good. You guys are on it. Thank God. Some dead people in here right now. I'm going to go around and take everyone's bills. Okay, so here's some fun facts about eukaryotic origins. Right, so there's many origins per chromosome, hence me not asking you how many DNA polymerases were associated with the genome during replication, because that would be psychotic. They tend to be activated in clusters, which are called replication units, which might contain 20 to 80 origins. They're activated at different times during the cell cycle, and we saw that in that paper that we did last week. Spacing of origins within replication units. So these origins might be anywhere from 30 kb to 250 kb, so 1,000 bases apart. And there are 2 replication forks per replication bubble, and they move outwards until they merge with another bubble. Right, so here you have the small replication bubble, and it gets larger, and eventually they're going to meet multiple replication bubbles, and then you have your whole chromosome replicated. Any questions? Does this seem like it's a big repeat from 309, or is it new? Okay, no one's saying anything. That scares me. Okay, so we're going to end with a short discussion with our friends. My friends, I don't know, your friends. So we're going to do compare and contrast. Regulation of initiation of DNA replication in bacteria versus eukaryotic cells. Discuss. It's going to be like coffee talk. Who used to watch Saturday Night Live in the 90s? None of you. Okay, so do you remember it, like Mike Myers, Wayne's World, Party Time, excellent. Thank you. Represent. Okay. So like he did this coffee talk, like old Jewish lady who would be like, I'm going to give you a topic, discuss. Anyways, discuss. Talk to your friends. I want you, this could be on an exam, right? I might say compare and contrast regulation of DNA replication initiation in bacteria versus eukaryotic cells. What I mean by regulation is what turns it on and what prevents it from being turned on. I don't mean what proteins bind. I don't care about that, but we're talking about regulation of initiation. So what regulates whether it's going to be turned on or not. So you've got like 8 minutes, and so we're going to end with that. So I'll give you a few minutes to talk, and then we'll discuss.
No.
There's always
They need to buy, they have to go to when they rent, they automatically have to go on and buy
Okay, guys. For the last 2 minutes, we'll discuss. So, what did we come up with? I'll call on random people. Charles Reed. I feel like he must be in the middle. I feel like I call him a lot and he doesn't answer. Where is he? Okay. Doris.
I said in material, have insulation of origin, and in material, just have like differentiated protein, just bind to the region, goes to the A to reach 0%. Also said like let's talk about polysaccharide. Do we have the polysaccharide periods?
Yes, we have them both. Okay, so let me repeat what she said. Methylation of origin in bacteria versus just binding of origin recognition proteins in eukaryotic cells. But there's a level of regulation there that you missed, but that does happen. But then there's what turns it on, right? That's the real question. And then what was the next one you said? One origin. So that's not really regulation. It's just a fact, right? So regulation, I'm really asking what turns it on, what prevents it from turning on. So, but you're right. Methylation's a big one, right? You have to have that complete methylation in order for initiation of DNA replication in bacteria. Yes?
Don't you need SCDKs in your replication?
SCDKs in eukaryotic cells, right? Remember, you've got that pre-RC, pre-replication complex, which consists of the origin. Mind you, I didn't teach you about this in great detail just yet, but we did go over it in the context of cell cycles, right? So this origin recognition complex just kind of sits there and isn't turned on until SCDK activates it. Yes?
So what about for batteries? Don't they have CDK?
Nope. No CD. I really want to say no CDKs in prokaryotes, but I feel the need to double check myself. Yeah, but no, it's not regulated in that way. Cell cycle is not regulated at all in the same way. There are a lot of similar things, but no. Yeah.
The bacteria only methylate their cytosines.
Do bacteria only methylate their cytosines? I believe they're not methylated cytosines in bacteria. No. They do methylate cytosines, but I believe it's not only that. Yes?
Is specific protein recruitment considered a form of regulation?
Is specific protein recruitment considered regulation? Yes, something that regulates whether or not a protein can be recruited is regulation, right? So that's, I think that they don't know. I'm giving away exam questions over here. I'm just kidding. But that's what I'm going to start doing now when people seem to start talking about it. Okay, get out of here.
No.